1 second. Okay. So yeah, thanks for joining everyone. Today we are excited to have Annalisa Marsika with us. She's currently leading a lab at Helmholtz Munich, and before that she was a professor at Freie Universität Berlin, and did her PhD at TU Dresden in Germany. And yeah, today she's going to talk to us about towards universal AI models for protein-RNA interaction prediction and RNA therapeutic design. So that's a topic that a lot of us at Mila are excited about and work on. And yeah, with that, I'll give the floor to Annalisa.
Yeah, thank you, Benno, and thank you so much for having me here. I'm very excited to give this talk. So yes, today I will talk a little bit of the research about the research that we do in our computational RNA biology lab at Helmholtz, and in particular in the field of AI for RNAs and RNA therapeutics, which is actually very exciting at the moment because RNA therapeutics are gaining a lot of momentum also in the pharma industry. So what I tell my students is always I've been working on RNAs in the last 15 years and all of a sudden they matter somehow. So it's really exciting.
Maybe just one thing before we started, I forgot to say, like, for questions, please feel free to put it in the chat or like raise your hands, and then we also have time at the end for questions.
Yes, you can interrupt me anytime. Yeah, it's actually really fine. We don't have to arrive to the end. So what we're interested in, or what is extremely interesting, is to try to see how AI can help us to understand RNA biology, and in particular processes that happen in the cell, which are called so-called transcriptional regulation control, which is whatever happens to the RNA life once it has been transcribed from the DNA. And what are the players in all the processes which lead to splicing, for example, to alternative forms of RNAs, which will give rise to different proteins, what controls the decay of RNAs in the cells, what controls the translation rates. So being able to understand these processes is extremely important for developing, for example, mRNA vaccines, because translation efficiency and degradation of such molecules in the body or in the cells of interest is crucial for these vaccines to work. And so one of the main players of post-transcriptional regulation that control the RNA fades are RNA binding proteins. So these balls here, these blue balls, and they are basically controlling every aspect from the birth of the RNA to its death. And whenever there is perturbation in some of the interactions between the RNAs and this protein, I don't know, for example, mutations or mutation in the protein or other RNAs, this has been found to be the cause of many diseases, including genetic diseases, but also metabolic diseases and cancer. So clearly, these players open up new therapeutic ways how to treat potentially these diseases. And they are also extremely interesting RNAs because they can be targeted directly, for example, with so-called antisense oligonucleotide drugs to inhibit their function, but they're also very interesting as diagnostic and prognostic markers because they can be found in body fluids, and by that they can enable, for example, faster diagnosis than a biopsy, for example. Just to give you a very rough overview, I don't want to give you details about how the RNA biology field has evolved so far, but I just want to give you an idea of the massive evolution that this field has been undergoing in the last year. And this is mainly due to the fact that around at the beginning of 2000, so almost...
There's one question in the chat.
Yeah.
The question is very simple. What is RNA? Someone is asking.
Okay, Alistair. Okay, so an RNA is so the central dogma of biology. Let's say it says that our DNA encode the genetic information to produce certain proteins in the cell, and this production of proteins is mediated by a molecule which is called RNA. So the DNA is transcribed, which means is read by some molecules to produce some polymer, some if you want a string, you can think about a string composed of 4 nucleotides, which is a polished version of the original DNA, and this RNA molecule will give rise to proteins because some machinery will go there and will produce a protein out out of the RNA. So this is the central dogma, but it became clear that RNAs themselves are not simple messengers of this information to form proteins, but they can work amazingly as molecules by themselves. And not the protein, but for example, the RNA itself was found to be, this was let's say, a breakthrough in 2005 when people discovered that small RNAs called microRNAs are better hallmarks or biomarkers of cancers than proteins. And since then, there has been a huge development of technology in the biotech field to measure different properties of the RNA. So the RNA is produced by the DNA, but then it's processed, forms alternative forms, can be degraded, forms some shapes due to interactions that can happen between the nucleotides that form. And we can measure things like, okay, how fast and efficiently can form a protein or will be degraded and cut out in the cell, which proteins actually this RNA binds, because this is crucial for its function. So if it binds the right partners or not. So according to the principle, tell me who your friends are, and this tells me who you are, basically. So we can understand a lot about the function of RNAs by the proteins that interacts with. But anyway, there has been a lot of development in techniques that can measure, for example, the structure of an RNA, how fast it's degraded, what binds, etc., etc. So there have been also a lot of consortia collecting and annotating these molecules when they were found in the cell or in patients, for example, in cell cultures, that now we have an understanding of how many they are in the cell. And there are hundreds of thousands, actually, way more than the protein repertoire that a cell can produce. And yes, and they enter the pharma. By that has diagnostic biomarkers, but also has therapeutics. And obviously all of you know what, or well, at least you heard what mRNA vaccines are, because there was this huge breakthrough during the SARS-CoV-2 pandemics, where there was, the technology was there since 20 years, but it was actually speeding up during this period. So RNA therapeutics includes mRNA vaccines. mRNAs are long RNAs, which eventually translate for proteins, but RNA therapeutics or RNA-like drugs include something called ASO, which stands for antisense oligonucleotides. And these are short RNAs that can go and hybridize, so attach to a long RNA, and by that inhibit its function or some other process. What is gaining a lot of momentum in the pharma industry is also small molecule that can bind to specific elements of RNAs. And to date, there is 11 FDA approved, for example, ASO's drugs, again, mRNAs. And the number of RNA molecules that enter clinical trials is growing and growing. So, but why RNAs are so interesting as therapeutic targets as well, not only as vaccines? Well, because our genome is mainly basically composed at 70% of non-coding transcriptome, which means those RNAs that do not produce any protein. And only the remaining part codes for proteins, but not all these proteins can be targeted by drugs. So only a certain fraction of enzymes, for example, can be targeted by classical drugs, the ones that we take every day. So in principle, the targetable proteome, that means what we really can use to produce drugs, is a very, very small percentage of what all our genome potentially encodes. And if we would be able to target directly RNAs and not the protein that they produce, or also those non-coding RNAs, in principle, we could target every gene in the cell. This is theoretically possible, right? And therefore, we could even hope to find treatment for diseases that actually don't have a cure due to technical limitation of targeting certain proteins. So machine learning model for RNA sequence and structure, we'll come to the structure in a second, include models to predict protein-RNA interaction. So given certain RNAs or a certain protein, can they form an interaction, and what is driving this prediction? splicing and translation, which are RNA processing. So the RNA gets cut, can form alternative forms. This is very important for the protein production and translation as well. One wants to find in these long RNAs functional motifs, which means are there parts of the RNA that really matter compared to others. We want to predict what function they have in the cell. So do they go and, I don't know, accumulate in certain organ upon diseases or whatever. We want to predict dynamics. We want to predict how they behave with other genes or so-called gene regulatory network in the cells. We can also screen basically for antisense oligonucleotides that are effective with AI. And ultimately, with the newest generative AI models, we can attempt to generate RNA in silico, which are better than endogenous RNAs, and use them as therapeutics. Our own contribution in the field in the last 10 years has been in the microRNA regulation, in the RNA-protein interaction tools, in trying to predict the function of these RNAs, and recently we are doing a lot of AI-assisted RNA design. So in general, our lab mission is to advance the understanding of this molecule function, their behavior in complex biological networks involved health and disease, and we want to both discover novel RNA targets, which means molecules that can be addressed therapeutically, but we also want to design RNAs which can work as drugs. And we do this with AI methods. So from sequence-based models, deep learning models, to foundation models for RNAs, which are more and more, and part of the lab also is heavily into graph machine learning for detecting these potential targets, which we'll not talk too much today. And just to give you a bit of context, why am I giving a talk today is because our Helmholtz and Mila and McGill and Université de Montréal have a formal collaboration into a framework called International Lab, where we allow an exchange of students from Germany to go to Mila and vice versa, inside a mobility grant, which is called Learning Causal Models in Single Cell Dynamics. And there are several PIs that were originally involved in Mila into this exchange, and these are the PIs involved on our side. And this is why we are having also this kind of exchange talk and mutual visits between the labs. Okay, so yes. So I will focus today on RNA protein interactions, on foundation models for RNA biology, and touch the aspect of RNA therapeutics. Okay, so how do we model protein-RNA interactions and why they are so important? Because understanding those interactions between these two different kinds of molecules helps us understanding why do we see some disease phenotype when something goes wrong in this process of the interactions. But also to model this process is important to engineer RNAs with desired properties, for example. RNA binding protein are important. This picture is just to show you that they are important for let's say, for every process the RNA is involved in the cell. One detour that I have to make is that when we talk about these polymers, these strings, which are the RNAs, we cannot avoid of talking about RNA secondary structure. That means that even if these are linear molecule, because of some biochemistry there, they tend to interact, to connect, to be attracted to each other, and they form something we call RNA secondary structure. This is just an example. The secondary structure happens because some hydrogen bonds form between G and C nucleotides and U and A, and sometimes weaker between G and U. And when we usually represent this for computer purposes or for our algorithms, is either a 2D representation, which highlights some main structural element, like, for example, they can form hairpin, they can form a stem like this, where the dots represent the nucleotides that interact. And when we have a 2D structure, which we can predict directly from the nucleotide sequence of the RNA, so if this is our sequence, we can actually represent it as a string of dots and brackets, where the dots represent nucleotides which are not forming any base pair hydrogen bond interaction, while the closing brackets represent, for example, the interaction between nucleotides. And then you can convert it back to a string, which is not the original string anymore, but it's a new alphabet that tells you, for example, this nucleotide is in a stem, and these are in a hairpin, and I will indicate it with H. And this is important when we want to encode this information in our algorithms. And then this, just for completeness, RNA binding proteins either like to bind to specific sequences of the RNA, or they like to bind to some particular shape. And people have found all kind of combination, and this is one of the main open questions in the field. So what is given a certain protein that I know binds RNA because of some particular domains, what's actually the preference of nucleotides? What's the pattern that is preferred by this protein on the RNA? And how do we measure? Where do we get data, basically, to build these models? One of the main biological techniques, it's called ECLIP, which stands for, or iClip, in vivo individual nucleotide resolution, UV cross-linking, whatever. This is just a technique that irradiates basically our cells with UV light, such that we can induce, whenever a protein is binding RNA, we can fix it, like we can induce some covalent bond. Then we take the cell, we chop the RNA, and we are interested in all these fragments that are bound by the protein. We pull them down, and then we try to sequence them. And after we sequence them, we end up with a library where basically our experiment produces some reads. Reads is like measuring the position at which the protein was binding the RNA at single nucleotide resolution. And those positions form some kind of cross-link events or interesting events that we want to model. So these events happen at particular sequences. So what our model do is we go from one experiment per protein, where we get the sequences basically where the protein was bound. We build a model that can predict basically where the binding of the protein, and then we go back and try to interpret the model and try to understand what was the sequence pattern or the sequence patterns that were driving the prediction. So just to give you an idea, this is like this coverage here represents how the experiments look like, and this is what you get, the footprint of the binding basically of the protein on the RNAs. And we have put a lot of effort in the past to try to understand how to find these positions from this massive data. You have to imagine, like, you have millions of sequences and you try to find all the points where this protein was binding in the cell. And so what we did in the past is like to take all these sequences, these peaks here, retrieve the underlying sequence, have a convolutional neural network with some pooling, which predicts basically for a certain region if this is bound or not bound by the protein. And what we did in our Pysster model is not only basically take the sequence underlying this binding and one-hot encode it, but convert it also to a secondary structure alphabet, which we used as combined encoding with the sequence. So if our sequence comes from an alphabet of size 5 and our structural alphabet is also size 5, then you have 16 possible combinations for your nucleotides, and this is our encoding. And this is how the model learns. And then basically, when we go to interpret it, this was a very easy interpretation method, like maximum activation of the kernels of the first layer of the convolutional network. Then you can deconvolute basically the sequence and the structure information. And the good predictive model will show, for example, for a certain motif, an accumulation in the positive class sequences at the center of your peak versus some basically uniform distribution in the negative class. Okay, so the way we did it was actually, we have at the end a 3-way classification model, because these experiments come with some technical bias. So how you choose your negative set is, I would say, the most crucial thing here. And the model works well, but in reality, it really depends how you choose your negative set. So for example, our positive set is the number of, or the sequences that are bound by the protein. If your negative set is, pieces of RNA, whatever, in the cell, then you might be learning just the bias of the experiment and not the specific of the protein. So what we do is here we choose negative set among other peaks bound for these RNAs, but not from our protein of interest, but from other proteins. Why we do that? Because the experiment has a preference for certain sequences, which is not biological. So by contrasting on a negative set, which is the sequences bound by other protein than the protein of interest, we are kind of having the same bias in there, and the model can focus really what's specific to your protein. So this was a very important point because at the beginning we were predicting a lot of nice biases. And since we had this tool, now we started wondering, like 2 years ago, or 1 year ago, what is the state of the art in RBP binding prediction models? And we did an extensive benchmark because since then a lot of architecture were explored. For example, STM, CNN, but also, for example, language model that could focus on this task, and also some shallow learning with support vector machines. And we established basically 3 very extensive data sets to benchmark the methods on. So to classify positives against 2 types of negatives. So as I told you, if your protein of interest is the red one, then you can have that your negatives are sequences bound by another protein or sequences that are unbound. And in the case of negative set 1, unbound sequences, your classification problem is much easier than type of negative 2. And yeah, so what we saw is indeed that the performance of the model for all methods and all these points correspond to about 130 different RNA binding proteins and 130 different models, actually. So it degrades immensely when you go from negative set 1 to negative set 2 for all methods. And what we also observed is that multi-label models, which predict not only if a certain sequence is bound or not bound by a protein, but they try to predict for all protein at the same time, at least the ones that use early stopping, because we have an outlier here, they actually outperform single label model because many proteins are very similar to each other, so they share some preferences, so this is shared among tasks, and this benefits the learning. And the other thing we learned is that we benefit more from input modality than from the architecture of the model itself. For example, if we don't only use sequence to learn, but we use also sequence conservation across species, and we use the genomic annotation of where the binding site is in my RNA. So it's at the beginning of the RNA, it's at the end of the RNA. This helps the learning. And also having a wider sequence context basically gives better performances. And then we wondered, so this is based on sequence only. Yeah? What if I encode the RNA secondary structure? Can I improve? And the answer is, when I predict the structure from sequence with certain tools, not really. You see that there is no real benefit of using this structure information for almost all protein. But when you can encode the experimentally measured structure, which is also coming from some high-throughput experiments of the RNA, actually you can really, for some proteins, improve your model. So there are some proteins that actually like to exhibit certain structure for the protein that binds. And the effect is still dependent on the negative set, being the task much more difficult for negative set 2 compared to 1. Okay, so another thing is like, well, why do we build these models? Actually, at the end, what we have in reality is we predict if an RNA is bound by a certain RNA binding protein in the training condition, but in biology, in practice, what you have is a new experiment on a new cell types, maybe a new organism. You don't have the data, you don't have this high throughput data, but you still want to know if your certain protein is binding certain RNAs. So we wonder to which extent models that are predicting, that are built in some specific context, can generalize to a new context, for example, to another tissue. And you can actually see that what happens here is that you can still generalize to a certain extent. Your performance degrades for those models which were not good to start with. So in principle, this cross-cell setting, cross-cell type setting is something that we can do. Okay, so after this work, we started realizing what are the limitations of these classification-based approaches, basically. So the classification-based approach that I just presented predicts whether an RNA is bound or unbound by the target. But in reality, the signal, the binding signal, is continuous. And it looks like the blue one here. So it's kind of a footprint and has a different shape for every protein. So when we decide, I don't know, this is a binding site, this is a binding site, but is this green one and red one really, we are really able to discriminate between these two? We take an arbitrary decision there. We put the cutoff and we say, okay, this is my negative, this is my positive, and the labels assigned to the binding site are really arbitrary. So such classification methods rely on the pre-processing of our data. So what we decide is a positive or a negative. And through this binary label, we also lose a lot of information on how the footprint of the protein works. So a lot of models in genomics gained momentum in the last year, which are some kind of sequence to signal models that try to predict not if there is a binding or no binding, but given my experiment and the sequences in this experiment, can you predict the footprint, so the shape of the signal? And this has been done not only for RNAs, but we pioneered it for RNAs. And so what we try to predict here is just the shape signal and not the absolute intensity of the signal, because this is really dependent on how abundant the RNA is in the cell. So we came up with a method which is called RBPNet and predicts protein-RNA interaction via sequence-to-signal learning. And this was the work was pioneered by one of my previous PhD students in the lab, Mark Horlacher. And how does it work, basically? So for each experiment which is available in public repository, we select 300 nucleotide windows across the transcriptome. The transcriptome is the pool of RNAs in your cell, okay? Based on some lenient count thresholding, and by that we train on up 1 million samples or sequences, which span most of the signal generated by the experiment. And then what do we do? Basically, RBPNet is a deep convolutional sequence-to-signal neural network. It takes as input an RNA sequence, and what it does, it outputs the probability vector of the same length, which describes a discrete distribution of your counts, of your reads from the experiment within that sequence. And in the CLIP experiment, basically predicts the distribution of these cross-link events as a result of the protein binding there, basically. And let's go here. Maybe the RBPNet architecture consists of two major blocks, if you want, or paths. So the model body, so the input layer, which is followed by several dilated convolutional blocks with residual connection, and then the model head, the output head, basically, which performs the final mapping, of the input sequence representation from the previous block, from the body model, to a probability vector. And during training, this predicted probability vector basically is used to parameterize a multinomial distribution of these cross-link counts, given actually the position-wide observed counts in the input sequence. And then a log likelihood negative loss is actually computed. So basically, the model is penalized in case it's unlikely that the observed cross-link counts are drawn by the distribution predicted by the model. So basically, then RBPNet, what does it learns the shape of the signal which is subjected to the RNA sequence under the assumption that is actually the RNA sequence, the driver that of the binding site of the protein. Another thing that RBPNet does is we know that the CLIP experiment contains a lot of experimental biases. We know that some sequences cross-link much easily in the cell than others. And this bias is sequence dependent. So we asked ourselves, can we model it? And this affects the distribution, basically. So every eCLIP experiment comes also naturally with a control experiments, which is kind of, you have to think about as a control signal, yeah, that you can get from your experiment, which is supposed to capture the experimental biases. So as we have this control experiment, then we can explicitly model it. And so to prevent actually learning biases, so the RBPNet model explicitly models the cross-link signal of the control experiment as well, alongside the real signal. And specifically, actually, RBPNet tries to explain this eCLIP signal by a mixture, so an additive mixture of these two signal components, the control and the bias. And the unobserved target signal. So basically, the real signal, you don't observe it, but you observe a total signal, which is the mixture of the two. So the total signal, which is telling us here P total, the probability distribution, the total probability distribution is given by a weighted sum of the two components, of the mixture of the two components, the target and the control. The control is the bias. And this is parameterized by a coefficient, a pi, which is more or less big depending how contaminated your experiment is by the bias. And so what at the end you have is, even if RBPNet is trained on sequences which are 300 nucleotide longs, the convolutional nature of the model allows you to input an RNA of arbitrary length and basically get your prediction. And this is just an example. This is how an RNA usually represented visually, where all these arrows indicate different blocks that form the RNA. And so this is your real experiment, the blue, and this is one beautiful example where, for example, the RBPNet prediction really recapitulates the original experiment. Obviously, they don't all look like that, but this is the idea. So what can you do once you can predict? Well, the interesting thing is most likely in the interpretation. So why have we predicted that? And we can understand why and which sequence one underlying. Maybe we can perturb it and we can do genetic engineering there. And so what we do is, like, as the predicted signal is a mixture of a control of a target, we can decompose the predicted signal, right? And use integrated gradients, for example, to interrogate the models and say, okay, what was the part of the sequence that was mostly activated for this prediction? And then one can try to understand what comes, like, genuinely from the real experiment of the protein and what comes, for example, from the bias. And we do this for all the sequences. We extract the important sequence or the explanation of the models with integrated gradients. We chop them into k-mers, we align them, and we build the consensus motif such that we can say, okay, for this protein, this was the main driving sequences. We do this and we compile a database of a catalog of RBP motifs for more than 100 CLIP experiments from the ENCODE. But the important thing is that this model can, for example, used to score the impact of genetic mutation on protein-RNA interaction. So if you have in an RNA a mutation which is deleterious, for example, and which drives certain disease, you can ask yourself, is due to the fact that the signal of the protein-RNA interaction is perturbed? And when you apply your model, in the presence of this mutation, you can have two different tracks. You can compute AKL divergence, and you can prioritize, for example, mutations that are important. And this is why this is important, because many mutations that happens related to diseases or not, they don't have an explanation. They don't have a cause at the moment. So molecularly, we don't know why certain mutation in certain RNA regions are leading to a disease phenotype. But when you can understand that it's because the binding of the protein is potentially affected, then we can do something for it. This is just other. So what are also these models good for? We can characterize RNA regulons, which means given a certain protein, which RNAs and which cellular pathways in the cells is addressing. We can do cross-cell line, cross-species, host-pathogen interaction inference, and we can predict a lot of other things. For example, one example is that we studied a protein which is called ROCK1, and it's a very, very important protein in our body, which binds to RNAs and to certain structures that look like this. So mutation in this protein lead to autoimmune diseases. Okay, so we did massive experiments with collaborators to produce these CLIP libraries to try to learn the preferences of these models. And we, for example, applied Pysster on the sequences and retrieved the structural preferences. And then we took the kernel, we computed the maximum activation back into the sequences. And we searched for sequence motifs into these sequences. And what we could derive is that there are certain K states for kernels, which encode certain certain structural preferences of your protein. And if they co-localize with sequence preferences, that gives you a repertoire of the RNA parts which are important for this protein to bind. And we also can take our model, for example, our RNA network, and trying to predict what SNP stays for single nucleotide polymorphism, so mutation. If I have a mutation in my RNA sequence in correspondence of the binding of the protein, is the model predicting that has a delta probability. So, and by that we can predict what are those mutations that will lead to a loss of binding, and also mutation that might lead to a gain of binding, actually, just according to the model. And then we give them to the experimentalists and we say, hey, we think these two mutations are very important for the aberrant function of the protein. And also the integrated gradients, when you do again the interpretation, can give you a hint, basically, if really this is affecting the sequence importance, the explanation of the models. And this helps you to rank, basically, this mutation. And then you can see if the perturbation of this mutation is really significant by doing an in silico mutagenesis screening of all the flanking position. And then you can see, okay, actually, I'm not in the background, the perturbation that I observed, the delta probability from the model is actually significant. And why is this so important? Is it affecting the structure of the RNA when the mutation is there? So is the base pairing or the shape altered? And this also can be assessed. For example, this is one case where this disrupting a U-rich stem loop by rocking led to the formation of a completely different secondary structure. And this helps you to really characterize those mutations which are important for the disease. So these models are also good for cross-cell line, cross-species, and host-pathogen interaction, because once you have a model on your protein of interest, which is been trained on a certain human cell lines, then you can apply and predict on another RNAs of arbitrary length in another tissue. You can also go across species. For example, you can predict in mouse, or you can predict on exogenous RNAs, for example, coming from a virus in a viral infection. And this is what we have done. We have applied this model to build an in silico map of all the human proteins that could potentially bind to the SARS-CoV-2 RNA. So SARS-CoV-2 is an RNA virus, so clearly exploits the human host proteins for its purposes, for replication, for survival, etc. So we identifying a group of proteins which, according to our models, were enriched on the virus, and we could also validate one of them. This led to a very nice publication from our experimentalist. Okay, so now our previous observation is that multi-label models of protein-RNA interactions seem to outperform single-label model. So our RBPNet model that I just presented is one model per protein. So it basically predicts the footprint of every protein singularly. But then and prompted by the results that we see for the classification model, we thought, can we extend this to all the protein for which we have experiments? So we take all raw data and we build panRBPNet, so a model that predicts, it goes from a single task to a multitask prediction model. So basically learns RNA representation for the bound RBPO, which means given all the possible combinations of binding of the proteins, can we learn RNA representation of this interactome, or this interaction space? Because if we can, then maybe we can use this learned representation to model several RNA processes. So this is still our work in progress, and panRBPNet is unpublished, but we extend RBPNet to learn on multiple RBPs at the same time. And so we have different output heads, basically. We also increased the number of parameters of our model to 70 million or so. And we also put a penalty on our mixing coefficient to improve the learning. And so does a multitask model improves in performance compared to single task models? Well, not dramatically, but yes, you can have usually an improvement in performance with these models, both in mean Spearman correlation coefficient between real signal and predicted signal, and also in the precision recall curve when you try to call binding sites on your signal. So actually, we observe the same, that also for this continuous model, we benefit from the multitask. And then we thought, okay, we have this model that somehow learns this representation. And nowadays, we have the advent of a lot of learned language models, also for RNA biology, so-called foundation models. And there has been a rise of papers in the field. And well, what a RNA language model does, it captures the sequence or the context of a nucleotides. So it's trained on corpuses of sequence in a masked like type of way to predict the masked part of an RNA or a DNA, like language models in general. And so this is just an example. It's a language model called RENALMO. So in the pre-training phase, you have a database of RNA sequences, you have some masking, and you have your RENALMO model with transformer blocks that learns to predict the masked nucleotides. But what really interests us in this field is that what we can do, actually, we are not interested in reconstructing at this stage, for example, the RNA sequence, but we are interested in what the learned representation can enable us. So, for example, if one takes the learned representation from RENALMO, can be used to do secondary structure prediction. So basically, it can be used to fine-tune for downstream tasks. So you can predict splicing, you can predict translational efficiency, you can use this representation to classify families or RNAs into different families. So we thought, okay, still language models are very powerful but they are really, really consuming to train. RENALMO, I think, is 700 million of parameters. So can we try to do something similar with panRBPNet and consider it a general model for downstream tasks? So at the end, our model learns to predict RBP signal, but what we are interested in is the embedding of the model and how we could use it. And we thought we could use it to model RNA processes as well. We could use it also for RNA classification. We could use it, for example, to score mutation better. And we could use it for therapeutic design as well. So let's see if this works. So we tried to fine-tune this general model, panRBPNet, on splice site prediction. So we took the original architecture and we applied it to, instead of predicting the protein profile, from the learned representation of this interactome, can we predict if each nucleotide is part of a splicing site, splicing junction or not? So we train a model, but then we change the head for the new task. And we see that actually the pre-training really helps to improve the prediction, that it does extremely well, and that it does better than the state-of-the-art method, which is called Splice AI. Yes. Is there a question? Okay.
I think not, no.
Okay. Then can we try to attempt another task? For example, can we predict ribosome profiling? This is another measure, another RNA process, which measure how good is the RNA translated. And also there, panRBPNet beats state-of-the-art methods. And when you look at the embedding of the method at the first quartile and the last quartile, you can see that there is a separation of sequences also in the embedding space. And yes, you take the embeddings, you stick your embeddings into some multi-layer perceptron or even a random forest, and you can already do much better when you learn a representation than, for example, a UTR language model, another model based on convolutional neural networks, and which are trained directly on RNA sequences. Instead, the difference from panRBPNet is that we don't train on blindly on RNA sequence, naked RNA sequences. We train only on RNA sequences which show some interactions in the cells. And this is still raw data because you just take whatever signal your experiment is giving you, millions of sequences, and you feed them into this model. We use it to do variance scoring, and without going too much into the details, we can show that we can do that very well. We can also use the embedding to classify, for example, different genomic regions of the RNA, and we compare it qualitatively to other language models, such as the nucleotide transformer, RNA-FM, DNA-BER2. And we can also do a bit better than RNA-FM when we try to classify RNA in different families, although nobody trusts UMAP nowadays, and one should quantitatively compare them. And finally, for the therapeutic designs, well, we have this model that can predict RBP binding tracks for, 130 or up to 200 RBPs at the same time. And there's been a wave of models in biology called experimental profiles foundation models, which are similar to panRBPNet and what I showed you, but they are trained actually not on sequences, but they are trained on sequencing underlying some experimental signal. And these experimental signals can be different, but just to put it in context, panRBPNet belongs to this category where you take tracks of RNA-seq or genetic variation among individuals, you feed them to the model to reconstruct not your sequence, but the profile of the expression of your RNA or the profile of the binding on the RNA. So these are a bit slightly different from classical language models, but they help focusing on that part of the sequence, which has actually a role in the cell. And so what we do is here, if you have an RNA, yes, with this gray thing here on top, and you want to know if I want to design a drug against these RNAs, like an antisense oligonucleotides, means a short sequence that blocks some important parts. And if I have an RNA of 30,000 nucleotides, how can I actually prioritize the design of this? Where should the RNA go and block? And you can run, for example, panRBPNet and trying to predict where certain proteins are putatively supposed to bind. We combine also other language models, and what we do is like we simulate the process of having a drug that can block a certain part of the sequence. So in a sliding window-like approach, we mask progressively part of the sequence. We compute the perturbation of the signal according to the model, and we rank these regions. So we answer the question, how would mimicking a steric blocking ASO, so such oligonucleotide along a transcript, perturb the protein binding profile? This allows us to prioritize certain sequences. And at the end, we have a full pipeline, which is called Gasoline, which combines RBPNet with another tool, Borzoi, which actually answers the question, how would mimicking an ASO at a certain position perturb not the binding, but the expression of the transcript. So we combine this in a pipeline where we filter basically and prioritize certain sequences to synthesize. So what is next? So I've showed you a model which is called panRBPNet and can be trained on 200 or whatever proteins at a time. But in reality, we have experiments or raw data for 200 proteins, but the proteins that can potentially bind RNA in the cells are up to 1,400. That means that we are learning a representation of the RNA which is biased towards the data that we have. So even if we don't have experimental data, can we still try to do something? Can we include protein? Can we extrapolate beyond these 200 protein where there is no clip training data? So Mark, at the time when he was in my lab, visited Joshua Benjo's lab, and they started thinking of how this could be done. So if we could learn simultaneously a representation of the RNA from our panRBPNet, but we could use a protein language model to learn a representation of the protein and ask ourselves, are these compatible? And if I train on a certain number of RBP, up to which extent I can extrapolate to similar or very different proteins, and can we quantify the uncertainty of these putative predictive interactions? And now they started doing, and it's taken over from other people in my lab, is really to use a language model and some interpretation technique to decide if a certain protein has an RNA binding potential. On the other hand, we would learn an RNA embedding or RNA representation based on our panRBPNet, and we would use concatenation or cross-attention to learn the binding. And as you can have exponential number of combination of how these two molecules could interact, we will use some prior knowledge, low throughput data to guide the learning. And so this is still, sorry?
There's one question in the chat. Could you please elaborate on which metrics are used to quantify the perturbation effects? You can also read it if you want to think about it. But
yeah, so of a mutation or
maybe
for a mutation is the norm or KL divergence, but also for the therapeutics, it's just the delta probability of the model upon the masking or upon the mutation, and kind of also a significance if this delta probability is random or is actually better than random. And to evaluate this, you compute an empirical p-value by mutating or masking or position, looking at the empirical distribution and see if your delta probability is significant.
Okay, I hope that. Yeah.
Okay, so this is the full model or the vision, if you want, because we are still at the stage that we are trying to learn, but this is kind of almost a solved problem. What makes a protein a good RNA binding protein? So if we can learn it, like using language model representation, fine-tuning on our experimental data about RNA binding proteins, and trying to understand how to do this cross-attention layer here. So this is the full vision, that at the end we would have a model that can encompass the full space of the protein-RNA interactome. So, and so this is basically more or less the conclusion of this part. And I hope I convinced you that these experimentally driven foundation models are equally useful, if not in some cases better, at predicting certain RNA processes. But the main advantage is that having to train a model with 70 million parameters versus 700 is a big advantage, because it's sustainable. And it's also kind of interpretable in a way, because we know that the embedding is driven by this combinatorial interaction. So we can go and interrogate and try to understand what proteins actually and what motifs were important to predict that particular task. So what else we do in the lab, what you can talk to me about, is we do with pharma companies antisense oligonucleotide design, not only based on RBPNet, but also based purely on generative models, and we compare. We do actually design of so-called 5' and 3' UTR regions, which are very important regions in an mRNA vaccine, because they are actually dictating how efficiently a certain vaccine will produce your protein of interest or your antibody, and how stable it will be in your cell. We are trying to use embeddings from our models, for example, panRBPNet, to understand functional exons in CRISPR screening in cancer. We are also in the field of therapeutics looking at small molecule and RNA interactions. It follows a bit this scheme where you have two joint representation of a small molecule and then RNAs, and you want to concatenate it and predict some binding affinities. This is a very challenging task because we are in a domain where the data are very scarce and very biased. So the idea is, how can I take different data sets with all their batch effects from different labs and build a model that can actually generalize to unseen molecules or RNAs. And I want to mention that we do also a lot of graph representation learning in our lab to identify new potential drug targets. We had a very successful collaboration with the lab of Jian Tang and Mila, and we have a paper which is undergoing the second round of revision in Nature Biomedical Engineering, and it's in bioRxiv, and you can check it. And this was basically applying the RBPNet algorithm, which was published in NeuroIPS 2021, to prioritize to graph completion of link prediction on biomedical knowledge graph, for example, to predict zero-shot drug treatments for diseases. And this worked very well against state-of-the-art approaches. And we do several modifications to the RBPNet algorithm to adapt it to biomedical knowledge graphs, and a lot of design choices that actually make it not only more efficient for graphs up to 5-6 million edges, but also meaningful from a biological point of view. And we apply to Alzheimer's disease, and we find some very interesting prioritization across existing drugs, which have been validated by expert medical knowledge. And this was a nice exchange where who from my lab visited actually Jian Tang's lab and partnered up with Cao Cheng and Sophie now to try to adapt RBPNet for biomedical graphs. And Sophie came to visit us, and we finished this project, and several people of the lab got interested and tried to apply it to other tasks, and this led to a very nice paper. And with that, I would like to conclude and thank my lab and especially the people whose work I've been introducing today, and the pharma companies for funding many of my PhD students and all the funding, including our international lab exchange with Mila. And thank you, of course, for your attention and for holding up so long.
Thank you. So yeah, if there's any questions now, feel free to either speak up or just type them. And yeah, otherwise, thanks for the great talk. So there's one question, I think.
Okay.
Feel free to also speak if you want, Etienne. Okay. I think I typed the question, I guess. Yeah.
Okay, so maybe I can read it as well.
Yeah.
Could you? Oh no, this you can write it out. Mm-hmm. What is scale of disease your targets from cancer to behavior? Yeah. So yes, we started. Developing methods for, depends also on the industry partners, right? So we started, it's easier to start developing methods for cancer because you have a lot of data available, but yes, we target also psychiatric disorders, for example. Viral infection we have been looking at a lot, and with our industry partners is since the pandemics, respiratory diseases and rare viruses, less understood viruses. And for Roche, for example, the focus is on neurological diseases. What is the scale of one protein or from cancer to behavior? One protein or several? Sometimes it's really narrowing it to one protein, like, for example, in autoimmunity, this rocking protein seems to be doing all the job and targeting like 1/3 of the RNAs in the cell. In some other cases, it's just a combination of more proteins.
Okay.
Thank you. My question, just to be sure, by probability, which probability exactly were you referring to? I was referring to the predicted probability from the model. If you have a sequence and you feed it into the model, then the model will give you, if it's a classification, it will give you the probability that that's a binding site. If it's a continuous model, it's a probability distribution, right, in a certain region. So it's a multinomial distribution, if you want. It's a multinomial probability distribution. So it's the delta of two multinomial probability distribution, which can be computed by, I don't know, KL divergence or any other measure to measure the difference between two continuous signals. It's always the probability of the model, yes. Or of the predictive model based on the sequences.
Okay. If there's no more questions, then yeah, thank you for the talk. And it's been great to see there's so much like collaboration between the two institutions happening. I was vaguely aware, but I didn't know about the extent of it. So yeah.
Yeah.
Okay.
Right.
Thank you.
Bye-bye.
Have a nice weekend.
Yeah, you too.